I forgot to hit the record button. So, all right. So we're going to start off talking about mitosis and meiosis and the main differences between these. So mitosis, we kind of hit on yesterday, but our main differences, mitosis is going to be one phase, where meiosis is going to be two phases. So we're going to have the cell dividing once, and over here we'll have the cells dividing twice. And so when we are looking at mitosis, this is going to form genetically identical cells, where over here we will have genetically diverse cells. And both of these are going to require the chromosome to double in the S phase of interphase. That's what's going to form the sister chromatids. And in mitosis, those sister chromatids are going, the chromosomes will line up in the middle during metaphase, and then those sister chromatids will separate. And so in the end, you will have two diploid cells. However, in meiosis, the chromatids don't separate until the second phase. In the first phase, we're going to separate the homologous pairs, and then we will separate the sister chromatids. And so this will give us 4 haploid cells. And what is the difference between diploid and haploid? Got 2 sets of chromosomes. We have 1 set of chromosome. And then this is going, and why do we only want 1 set of chromosomes in our haploid cells?
The egg and the sperm will combine.
Correct, because this makes gametes. So then the egg and sperm will combine to make your zygote and give you a new diploid cell. And so the product of mitosis are going to be somatic cells. And the other easier name for somatic cells are the body cell. So everything else is a body cell. Just your gametes are going to do meiosis. And this is true for animals, some plants, not true at all for fungi. It's a completely different reproductive system. And then it doesn't apply to bacteria because they don't do sexual recombination like this. So our main differences, we're going to focus on just a few of the phases that are most important. So in prophase of mitosis, the main thing that is important is the condensation of the DNA into the chromosomes. And at this point, each chromosome is made up of the 2 sister chromatids. Yeah.
So normally the DNA are not condensed to chromosomes like when the cells are just by itself.
Right, because when they're condensed, you can't do gene expression. So then the DNA has to unwind so that you can have transcription. In meiosis 1, you also have that. But what else is going to happen in prophase 1 of meiosis? That's very important. Crossing over. In order for crossing over to happen, what has to occur first? Not fusion of the cells, but
Oh, they have to line up.
you're going to have synapsis, which is the pairing of the homologous chromosomes. So last year's AP test, there was a question about this, except a stated it wrong. They said in order for in phase one, chromosomes have to pair together in the process of crossing over. And if that doesn't happen, then, and so like they used the wrong term. They should have said synapsis, which then led to crossing over. And so all the students came back and they were like, they said it wrong, it wasn't correct. And we were and like, well, what did, and I was like, what did you do? Well, we answered it just like it was synapsis. I'm like, okay, good. So you answered what they were looking for. And then I wrote a polite but very pointed email to the college board and got a generic, please have all blah, 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 call this number. I'm like, you don't care.
Did you call the number?
No, because I knew it would go.
You know,
So I don't know if they did anything about that, and tossed that question because it was technically wrong, or if they just. So, all right, mitosis, metaphase, how are the chromosomes going to line up?
In a line.
One line or two lines?
One line.
One line, and when they are in one line. Along the metaphase plate, how do the spindle fibers attach?
To the side.
It's going to attach to both sides. And so that will result in what happening?
Breaking up sister.
That's going to separate our sister chromatids. Hello.
sorry, Elaine.
So then in meiosis one, how are the chromosomes going to line up?
Like
Two lines.
They're going to be in two lines. And how will the spindle fibers attach?
Only to one side.
And so when they separate, what happens? What do we actually separate? Correct. We separate the homologous pairs. When do the chromatids separate?
In the
Second.
anaphase two.
Correct. In the second phase, which is just like mitosis. So when we look at this, this is also going to be important because half of the set of chromosomes is from this individual's mother, and the other half is from the father. And do they line up all the mother on one side and all the father on the other? No, it's random. And the random lining up of your homologous chromosomes, that is called independent assortment. Which is going to be important because when they line up, the next time meiosis happens, they will line up differently. And the next time they line up, they'll line up differently again. And so that's going to give you a lot more genetic diversity in your gametes. Independent assortment.
And for mitosis, it's not.
Mitosis, that doesn't happen because they all line up right in the middle.
Like, Mr. Wynn, so homologous chromosome is just two chromosomes, like basically sister chromosome.
So we'll get to that. So, all right. So then how does meiosis and sexual reproduction cause genetic variation? So first of all, crossing over. When is crossing over actually going to matter? When the alleles are what? Different. If you have homologous genes and you have crossover, is it going to give you any different results?
No.
No. So when it's the genes are heterozygous, that's when crossing over is going to matter. Yes.
When it crosses over, this may be a dumb question, but will it cross over at the beginning and end of one gene or just never end?
So it could, it's not going to be, it shouldn't be random. It should be controlled. So everything lines up just right. So the crossover happens at the same spot on the chromosome. So even if it was somehow in the middle of a gene, it shouldn't be. It would still, you'd still get two whole genes,
Okay.
and wherever the mutation was that causes the different allele would then potentially be switched. But you could cross over multiple genes at a time.
Yeah,
Um,
but it's not gonna like affect a gene's the level, like it won't.
But it could, you could have crossover errors. And so that could delete part of a gene, that could duplicate genes, that could invert them, or the wrong two chromosomes line up and you get translocation errors, what's that called? So, all right, so then down here, we have our independent assortment, as we were just talking about. And so we've got these 4 chromosomes. They could line up like this, or they could line up like this. And so this gives you different gamete possibilities. And this is what we are doing when we do a dihybrid cross. And we are doing that FOIL method to determine all of our possible gametes. That's what that represents, is that independent assortment. And then, of course, you have the random fertilization. So when the males are making those billions of sperm, they are making all those different possible combinations of gametes. And then whichever sperm actually fertilizes the egg. And then when reproduction happens again, you have a different sperm fertilizing a different egg, and then that creates more genetic variation. So
So crossing over happens and then after that, they are also like assorted in a random order too.
yes. So your basic statistics is 2 to the n, where n is your haploid number. That's going to give you all of the possible numbers of gametes without crossing over. Yes.
So yesterday you said fraternal twins is just two different sperm. And
2 different eggs. So just like,
what about identical?
so then that's where you start with 1 egg with 1 sperm. And then in the early developmental stages, the cluster of eggs that is making the embryo splits. And so then those are genetically identical. And then those will develop in a single placenta, whereas fraternal twins will develop their own placentas.
How does that work with this? Like, does it like go out then?
So when the cells are dividing, and it forms a ball of cells, the outer portion of the ball forms the placenta. And then there's a cluster of cells on the inside called the inner cell mass. That's what develops into the embryo.
Oh,
So that's the part that's going to split into 2 to give you identical twins. And so then they,
so they're like in the same chamber.
yeah,
Oh, okay.
Is it possible to have identical triplets?
it is, but that usually, so 2 things could happen. It could divide, and then one of them divides again, then you get 3, or you had it divide twice and you had quadruplets and one of those embryos wasn't viable. So
So do you think if there's like, for example, identical triplets, do you think one of the triplets would be more developed than the other 2, or they just-
potentially, and that can happen even with identical twins, one is more developed than the other. So depending on where they're located in the uterus. So, okay, so yes.
That being detrimental for individual,
What?
genetic variation.
It can be genetic variation. It can be problematic because if you get the bad genes, you know, genetics is a lottery. Sometimes people get the good genes and sometimes you get the bad genes. And let's not use the word bad. That sounds so judgmental. You get the less effective genes for evolutionary success. All right, so when we look at this, we've got 2 homologous chromosomes. And when we say homologous chromosome, what do we mean? They have the same what?
Same gene.
function?
They have the same genes. And what is a gene?
Coding for a certain.
Correct. DNA segment that codes for a particular trait. Could be protein, could be an RNA, could be something. All right, and then what is an allele versus a gene?
Copy,
It is a, what do you mean by copy?
it's like a certain copy, there's like multiple different
Certain
variations of
phenotypes.
it.
Wait, sort of. So in meiosis, why do you call it homologous chromosome since they are not the same anymore after crossing over?
They're still homologous because they're carrying the same genes. So we see here, this is the gene for flower color. But they're different in that they have ones for a different color. So homologous chromosomes doesn't mean that they're genetically identical. It means that they're carrying the same genes for the same characteristics.
So the
But the
same trait,
characteristics could be different.
so it's the same trait, but not.
So it's the same gene, but not necessarily the same trait.
Yeah.
All right, and then the locus loci, so you'll see this sometimes. This just means the location of the gene. So if you see that on the AP test, and they're talking about a gene locus or a gene, the loci of the gene is just its location. And so this is this particular gene's location. This gene has a location, this gene, and they all have a specific spot. And there's a whole nomenclature in the scientific world about how to identify a gene's position based on its which chromosome, which arm of the chromosome, and then where it's located on that arm. When we talk about homologous chromosomes, we can see this. This is a karyotype. This is simply just a picture of an individual's chromosomes. And we see that these two chromosomes have the same length and the same banding pattern. That's what makes them homologous. The banding pattern is from stains that they use to stain the chromosomes. And so here we've got 22 chromosomes, and then you've got the 23rd chromosome pair, which are the sex chromosomes. And so all of these are the autosomal chromosomes or the body chromosomes or the somatic chromosomes. And then you have the sex chromosomes, and you either have the XY, which would be
Male.
and then XX for female. Sometimes you will have something called non-disjunction. And non-disjunction is when you have a failure of what to happen.
Separate.
Chromosomes don't line up in this one.
Right, so the chromatids don't separate, and so you might get an extra 21st chromosome. And what would that be called if you have an extra 20? That is Down syndrome or trisomy 21. You could potentially have someone who has monosomy, so that would be trisomy, an extra chromosome, or maybe you have only one X, and that is monosomy. That would still, this is still developed as female, because remember what happens to X chromosomes. One of them always
Turned off.
gets turned off and making a bar body. But you could have XXY, you could have XXX, you could have XYY. YY would not develop because you need at least one X chromosome for proper development. Yes,
It's kind of a weird question. I was reading a book, this girl was talking about like hermaphroditism.
right.
How did that develop?
So not going to be on the AP test. We'll go through this real fast. So hermaphrodism is when potentially, so no, everybody starts off as female, and then the ovaries, if you are genetically male, are so they are normally going to differentiate and develop into testes. And so sometimes you'll have one ovary that does that and one ovary that doesn't. So then a person can have one ovary and one testes. Or sometimes it's a partial, where it's part ovary, part testes. Or maybe they're genetically male and they don't develop into testes at all. And so you can have all of those different possibilities. So biology, despite what political affiliates may say, biology is not binary in sex determination, because you have this whole spectrum of different things that can happen. So, and then of course, psychology is an emergent property of biology. So if the biology isn't clear, then the psychology probably isn't clear either.
Then
So
you can impregnate yourself.
you cannot do that. So, all right, but you can have people who look genetically male and have a baby. That has happened. So,
Why do you only see trisomy 21? Why not with the other genes?
so there are some other trisomies that exist, but a lot of them aren't viable,
Oh, so
and they
it's
don't
overrated.
develop. They'll have spontaneous abortions, or they'll have miscarriages, things like that. All right, so when we talk about genetics, we talk about mutant types and wild types. So the wild type is what you would normally find in nature. And then the mutant types are the things that are different from that. So if we look at these two fruit flies, the wild type is this yellow body with normal wings. The wild type, or the mutant type over here, is the black body and the little shriveled vestigial wings. And so then we remember Thomas Morgan said, let's have a whole new system. And so we base the fruit fly system on the mutant type, and then we put the plus to represent the wild type. And so if you see this on the AP test, they love fruit fly problems. The plus is the wild type. The non-plus is the mutant type. They usually, and should, if they're doing their job, give you a key to explain that, but just in case they don't. So quickly, if we have a monohybrid cross where we're doing one trait, if we have feathered legs, capital F, which is dominant over non-feathered or clean legs, lowercase f, a heterozygous hen, so that means that she is
Yeah.
big F, little f. If that is crossed with a clean shanked rooster, so if he is showing the recessive trait, he has to be, and so then we do our cross, big F, little f, big F, little f, little f, little f, little f, little f. So then our genotypic ratio is 2 to 2, and our phenotypic ratio is 2 to 2. So if we have 12 eggs, and if all of those eggs hatch, what would we get?
About 6 and 6.
About 6 and 6, correct. They typically do monohybrids and multiple choice questions. Real quick cross, pick out the right answer. And then dihybrids, they might do in multiple choice, or they might do them in the free response. But what you will, what you should not have to do is a full dihybrid cross. Although they might, even though they have never seen it, you never know. So make sure you know how to do a full dihybrid cross. So that's where we're doing 2 traits. So if we've got capital Y, capital Y, capital R, little y, little y, little r, yellow is dominant over green, round peas over wrinkled peas. If you cross 2 of those, the only gametes that this will make is what?
Capital Y, capital
And the only gametes that this will make is little y, little r. So they cross and you get a heterozygote for both traits as the F1. That's if everything is homozygous for both traits, easy, you're just getting one type of gamete. But if it's heterozygous for both traits or even for one trait, then you're gonna have to do your FOIL. So big Y, big R, little r. So then my possible gametes, the first two will be big Y, big R, the outer two, big Y, little r, inner two, little y, big R, outer two. And those are your 4 possible gametes from that kind of cross. Statistically then, what is the probability that you will get any one of these gametes?
1 in 4,
What percentage is that?
25.
So that's going to be a 25% chance for any one of these. And then if you are crossing this with another big Y, big R, little r, what? The X is the cross. We're crossing those two. That's the wrong people. And so we're crossing those two. If you have 2 individuals that are heterozygous for both traits, and you have independent assortments, meaning they are not linked on the same chromosome, your ratio will be 9:3:1. That almost always shows up somewhere on the AP test. It's usually in the multiple choice question. So you just have to get that in your head. Heterozygous for both traits, independent assortment, or the language could be not linked on the same chromosome, then you will get a 9:3:1 ratio. It may not be asking that particularly. It may ask you if given, 64 offspring, how many of them would show both dominant traits? That would be the 9 right there. So it will be beneficial for you to do a full dihybrid cross, which is one of the homework problems. And so just so you work it out and you practice that, yes.
So if our ratio is like 18:6:2, like, do they want you to divide it to make it? Like that?
No, it's just, they might give you a number of offspring, and then you just have to make the connection that it's,
The genetic ratio is like 3:1 or 2:2.
I would put 2:2, they would accept 2:2, or they'd probably accept 1:1, but
No.
if it's a monohybrid, it should add up to 4. If it's a dihybrid, it should add up to 16. So, all right. So if we are doing this, you might be given a problem. And of course, we know that we can do everything as a probability. So this gives you the opportunity to take some shortcuts. So if they ask for the probability of getting big Y, little y, big R, you can do the entire dihybrid cross and count up how many of those there are out of 16. Or we can just use simple probability, knowing that big Y, and we just do our simple monohybrids. And we do. So with this cross, big Y, little y, big Y, little y, what's the probability of getting big Y, little y?
1/2.
It's 1/2, so we can get it here and we can get it there. So the probability of getting this is 1/2. And over here, what's the probability of getting big R, big R?
About 4.
And so then the probability of big Y, little y is 1/2. The probability of big R is 1/4. So then my total probability for big Y, little y, big R is
1.125.
1/8, yes. Which you can express on the test as a decimal or as a fraction. Both answers would be acceptable. So you can use these probability mechanisms to do your genetics problems faster. But if they are asking this on the free response test, and I'm going to say this to some people in very particular, you must show your work. So, all right. So when we do crossing over, again, crossing over only matters when it is heterozygous. So if both of these were big A, big B, if crossing over happened, you'd get the same result. It wouldn't matter. So when they are heterozygous and they cross over, the important thing to see is that some of the gametes that result have the normal parental types. So instead of normal, we have the gametes that are the same as the parents of this individual. So this individual right here, so this is one cell that's undergoing meiosis. This came, let's say, from the father, this came from the mother. And so the gametes that are produced, this is like its father's gamete, this is like the mother's gamete, but the crossing over gives us a recombinant gamete. And so when you have crossing over, when you cross this with another organism that is going to be homozygous recessive for both traits, it will then show us parental offspring and recombinant offspring. So here is, this is our heterozygous individual, big B, B plus, VG, VG plus, and then here is our homozygous recessive individual. This one is going to make what kind of gametes? If it's homozygous recessive for both traits. Right, it's going to be big B plus or B plus and VG. That's all it's going to make. This one, however, can make 4 gametes, big B, VG, B plus, VG, or we can do the crossover and get those, and that would be our recombinant type. And so when they reproduce, we see that you get some offspring that look like this parent, some offspring that look like this parent. That would be our parental offspring. And then we get the ones that don't look like the parents, because we've got the gray with the vestigial and the black with the normal wings. So these are the recombinant offspring. Now you will get this in normal independent assortment as well. However, the ratios of the parentals and the recombinants will be different. If you have independent assortments, what is the likelihood of getting the 4 different gametes? It is 25%. So you would have even distribution of these types of offspring. But if you have crossing over, since crossing over only happens some of the time, you'll get a higher percentage of your parentals and a lower percentage of your recombinants.
Why?
So independent assortment is even random distribution, but crossing over only happens a smaller fraction of the time because it just does. So, and so that'll affect the distribution of your offspring. And then we have to look at statistical tests to try to figure out how to do that, which we need to go really fast on. All right, so if we are looking at this, what we see is that these highlighted ones have a lower percentage, and these are the ones that are different. So these are our mutants or our recombinant offspring. And then these are the parental offspring, and our total number is 400. So if we see that our mutant or recombinant offspring are a much smaller percentage, we can use that to calculate our recombination frequency, which means how often those things cross over. So in order to calculate our recombination frequency, what we are going to do, we're going to take the total number of the recombinants. In this case, that is 72. We're going to divide that by our total offspring. So that is going to give us a number of 0.18. So that is the recombination frequency. So what that tells us is they cross over 18% of the time. the farther the genes are apart on the chromosome, the more often they will cross over. If we had equal distribution, if these numbers are about all the same, so if they were all about 100, that would be an indicator to our brain that these are not on the same chromosome, but they are independently assorted. So if you looked at your recombinants over your total and it was close to 50%, that means that they are independently assorted. We can then use recombination frequencies for multiple genes to draw a gene map. So what do we start with our gene mapping? Which number, which combination?
I think we just want-
Right, I'm gonna start here, the ones that are farthest apart. So A and C are 27% different, or 27% or 27 map units. That's just a relative term for the unit of how far they are apart on the chromosome. And so where would gene B be located? Closer to A or closer to C?
Okay.
A, because A and B are 10. This is not the scale. And so then if you had more genes, then you would have to puzzle piece all of that together and figure out their order. So looking at this with probability and some statistical methods, again, if we have independent assortments, that means that the genes are on separate chromosomes. If we have linked genes, the genes are on the same chromosome. So that means that big B and VG are going to be inherited together most of the time, where over here they sort independently. So this is where you would do FOIL. This is where we would expect an equal distribution of about 25% across the board. I don't know why that 2 keeps disappearing. Because as these sort and you get those 4 possibilities and you're combined, and then this is only giving you 1. So then you get these 25%. If we have crossover, and what we see down here is we have a higher percentage of the parental types. And then we have a lower percentage of your recombinants. And so what we can do is we can look at this data and we can figure out, is it actually due to crossing over, or did the random sorting happen to come out this way? And so if we are testing for randomness, what do we use? We're going to use a chi-squared. And this kind of problem shows up very often on the AP test. Testing for randomness of some way, whether it's for genetics like this or some other test. The way this looks in genetics is our null hypothesis, or randomness, means independent assortment. They are on separate chromosomes. If our chi-squared says it's not random, then we're rejecting the null hypothesis and we are saying that those genes are linked on the same chromosome. So both of these equal a total offspring of 200. So if it is random, the expected number would be what? Yes. So 200 randomly distributed into 4 categories, it'd be 50. We got to go fast, people. So then we do our chi-squared. When we do our chi-squared, we do observed minus expected. So 54 minus 50 squared. And then we do 46 minus 50 squared. 48 minus 50 squared. 52 minus 50 squared. We're going to add all of those together. And then we're going to divide it by the observed, which is 50. So 54 minus 50 is 4, square that, that's 16. 46 minus 50 is -4, squared is 16. 48, that's 2, so that's 4. 52 minus 50 is 2, so that is also 4.
Plus 8.8.
So that's gonna be 32 plus 8, which is 40. 40 divided by 50. We're coming over here for the equals, so that's 0.8. Coming down here, we're gonna do 63 minus 50 squared, which is 13 squared is 169. 38 minus 50 squared, that's 12, so that's 144. You would all do this on the calculator. I'm just remembering this from yesterday. 37 minus 50 squared, also 13. And then 62 minus 50 squared, also 12, 144. We're going to also divide that sum by 50. The answer to that, I don't remember. So 169 plus 169 plus 144 plus 144 is 626. Divided by 50. It's 12.52. So now so when we look at this, we now look at our confidence chart. How many degrees of freedom do we have for both of these?
3.
3. We have 4 categories. Degrees of freedom is categories -1. So we are looking here, degrees of freedom of 3 at a 95% confidence level, the 0.05 p-value. And so our value is 7.82. So the first one, 0.8 is less than 7.82. So this one is random. So that would be independent assortment. And then this one is greater than 7.82. So this would show we reject the null hypothesis, and this would show linked genes. Okay, so I'm gonna pause the recording, and then sometime later today, well, I'm just gonna stop it, and then I'll do a part 2. Put the part 2 on my Mailer.